Thank you, everybody. It looks like a more intimate meeting this time, but many people from genomics, for Jacob, just to let you know. Thank you for coming to present here. Good evening, everybody, and I hope more people are coming in. And enjoy.
All right. How do I advance the slide? Okay. Hi, everyone. My name is Jacob Oppenheim. I run R&D, data science, and machine learning at Indigo Agriculture. So a little bit about myself, to introduce myself. I have a doctorate in physics and biology. Before working at Indigo, I worked in genomics. Actually, my first boss is over there in the audience. And hi, Daniel. And since I've been at Indigo for about 2 years now, I've really built out our data science and machine learning platforms and how we think about this. So a little bit about Indigo. We are a plant microbiome company. So we are developing new microbial treatments for plants to counter arbitrary stresses in arbitrary agricultural crops in arbitrary geographies. Our headquarters are here in Boston, actually in Charlestown. Our commercial office is in Memphis. We have international presence in Australia, in Argentina, and in Brazil. When I joined back in November of 2015, there were about 30 people. Today, there's over 200 people. We have products in cotton, wheat, corn, rice, and soy, which are the major row crops. And so we're well on our way to actually transforming agriculture. And as of just recently, we've raised $300 million. So, what does data and genomics have to do with this? I believe that the plant microbiome has the potential to drive the next revolution in agriculture. And historically, since the Second World War, technological advances in the pharmaceutical and biotechnology industries have driven dramatic changes in agriculture. So for instance, the discovery of antibiotics led to the agricultural chemicals industry, and really our antifungals and other pesticides that we apply. Today, the therapeutic biotech revolution led to agricultural biotechnology, most especially genetic modification of plants. Now, we believe that the plant microbiome will yield this next revolutionary step change in agricultural yields. And this is because the microbiome allows us to target products between the genotype or variety of the plant, the microbe, stress, and the environment. So what this means is that if you are a farmer growing cotton in northern Texas where it's very dry, we can create a product to increase your yield and increase your drought stress. Whereas at the same time, if you are growing cotton in the upland south where you get lots of water and your yields are very high, but you're worried about cold and wet emergence because you might get too much rain early on in the season, we also have a product for that. So we believe the microbiome allows us to develop these products. So the point, though, is that we need to generate a lot of data if I have a microbe in my freezer to figure out where it will be beneficial and where to target this. The use of machine learning is to take all this data together and to allow ourselves to predict when something's just in the freezer and we have something just like a sequence, where it should be a product and how it should be. So we know from the literature that plant fitness can be profoundly influenced by microbes in around the plant. So hey, okay, all right. Hey, Eila, keeps flashing in and out.
I think it should be good now.
Oh, okay, thank you. Okay, so there's a long literature about various microbes that are known to induce insect, fungi, bacteria, virus, plant stresses against weeds, and also not only in biotic stresses, but in abiotic stresses, drought, cold, heat, salt, etc. There are nice stories around, for instance, a microbe that induces drought and heat resistance in Christmas cactus that grows in Arizona. And basically what happens here is that there's a fungus that 40% by weight of this fungus is this compound monocyllin. What monocyllin does is that it inactivates a certain heat shock protein in the plant, which stops it from killing itself when exposed to really high heat and drought. Similarly, there are microbes that allow plants to grow near geothermal springs in Yellowstone National Park. So there's this whole long literature around abiotic and biotic stresses and plants and how they can be alleviated by various microbial treatments. So the question then, again, becomes how do we take a microbe in the freezer, just get a little bit of information about it, either through sequencing or for assays, et cetera, and predict where it can be beneficial in the field and in what cases. So we've built an entire R&D pipeline around this. I'm just going to briefly sketch this here. We're going to focus on the left half of this, which is really where the machine learning comes into play. So we have tens of thousands of microbial samples. These come into us from sourcing in fields and through academic collaborators and other sources. So we can sequence microbes. We can do community sequencing experiments where we look for marker genes. And we can collect all of this information. We then take this information and use our scientific knowledge to predict what things will be beneficial. We pass these through a set of high-throughput screening assays before moving them into the greenhouse, eventually into the field. And then those microbes that succeed in the field eventually become commercial products. We monitor their success in the marketplace and use that as further data as sort of a validation step to really understand what we've learned. That makes sense? So when we collect microbes, one of the paradigms that we use is we go out to stress fields and we collect healthy plants under a target stress. This actually, as I believe, under cold wet emergence, the ground got very wet and then dried out, leading to this cracking. So you can see that there are larger, healthier plants and smaller, less healthy plants. So we can isolate microbes from both of these plants, and we can look at what's differentially abundant between them. The key technology here is community or amplicon sequencing. So when you have a microbiome sample, one thing that's common to do is amplify certain marker genes. This is the 16S region of RNA in bacteria and the ITS in fungi. Basic idea is you amplify these sequences and you sequence all of them and you know, and you use this information to inform you who is in your sample. So on the left-hand side, you can see sort of a demonstration of how this is done. And on the right-hand side, you can see the sorts of abundance measurements that we get. We can then do some computational prediction based off of this information. So we have thousands of microbes that we collected in sequence. We have hypotheses about how these might drive differential success in the field. And we can do some machine learning on this. So our pipeline portfolio is driven by hypotheses and supported by large amounts of data that we collect and generate. So one thing that you can do is that you can leverage the data you've collected as well as public data. So there's a lot of microbes, both bacteria and fungi, that have been whole genome sequenced that we know an awful lot about. And we should be able to use the information in public data to allow us to infer something about the phenotype of microbes that we've collected in the field. Furthermore, if we have performed assays or run tests on some subset of them, we should probably be able to infer the properties of their relatives in the strain collection that we have not yet tested. Now, if we do something like this, we can then nominate from our culture collection lots of strains that we may not yet have tested under the hypothesis that they will have this feature that we like. So essentially, this comes down to this question of how can we infer phenotype? And for the rest of the time, I'm just going to be talking to you about how we use some nice machine learning, genomic data, to infer and predict phenotype of strains. All right, I'm going to pause there if there are any questions. Yes?
Why do you find yourself sometimes? You have microbes that help animals too.
Right. There's plenty of companies that are trying to improve human health by modifying the human microbiome. Our focus is on improving agricultural yields under stressed and unstressed conditions, and so we're looking at the plant microbiome. Yes?
Do you use the entire genome or just some specific portions?
Right. So when you have a sample and you want to see what's in it, this community sequencing paradigm is like the most standard thing you can do in metagenomics. Is you just amplify this said barcode sequence that reflects, or it should be unique-ish among different strains or taxa of bacteria and fungi. It's not really, but it's pretty close to it. And that tells you what's there. You can do whole genome sequencing on a microbial sample with lots of things in there. The problem is that it's very hard to assemble the results and then back out what you actually found. So it can be useful functionally, but not at telling you who's there. When we do have individual microbes, however, we do whole genome sequencing to infer as much as we can from their genomes.
So how much, in terms of the plants, if you look at rice, right, there's a lot of varieties of rice.
Yeah.
So why would one particular fungus affect one variety of rice the same as another? You have to figure out what variety and what particular rice.
Right, so that's a really good question. There are going to be genotypic differences in responses of plants to microbes. This has been demonstrated in the literature. A lot of times, however, the genotypic differences are perhaps not all that large and perhaps reflect environmental differences. So for instance, if you look at how plant seeds are distributed in the United States and what's grown where, there's actually a long history of breeding plants that perform best in certain locations, especially in corn. This has been going on for well over a century now. So there's a specific set of varietals that are grown in specific locations, and this is really micro-targeted. You know, northern Illinois versus southern Illinois we're talking about here, right? So you can almost at times think about genotype of the plant as a function of the environment. Now, I'm not saying that's always going to be the case, but it's uniquely tied to that because when you optimize a crop for a specific region, you're in many ways influencing its genotype in that specific region. Does that answer your question? Yeah.
Do you ever do things like directed microevolution of bacteria?
Not yet.
Can you repeat the question?
Yes. Do we perform directed microevolution of bacteria? No. There's a long list of reasons around that we can discuss offline. Okay. Any other questions? Okay. Let's talk about some machine learning. So if we're going to infer phenotypes, if we're going to do some type of prediction of phenotype, we need to have a metric. We need to have a way of relating microbes to one another. So one question you can ask is, how do you represent, how do you ordinate the vast diversity of microbial life? So the traditional answer that you probably learned in high school biology was that we have this great taxonomic system devised by Linnaeus in the 18th century. You know, you have 7 set levels, and you can characterize things according to levels, and you would say, okay, things are more likely to be conserved at the genus level as opposed to the class level. Great. The problem is that the Linnaean system is very brittle. It's hard to jam in macrofauna like mammals that we are used to and we see every day, that we understand their morphology extraordinarily well. It's still hard to fit them into the Linnaean system. So imagine taking the vast diversity of bacterial and microbial life and putting them into the Linnaean system. You have 7 set layers. Is 7 the correct answer? How are we supposed to ordinate the continuum that is evolution? It's also ill-suited for microbes in the sense that it's very hard to define terms like class or order across all of microbial life, or even to compare across microbial life to non-microbial life, to eukaryotic life, for instance. So defining these terms in a useful way is, in fact, quite difficult. So you might say, okay, well, let's look at some genetic difference between strains. You might say, okay, if you have 90% similar genome, we're going to call you the same genus or something like this. So you can then say, I'm going to choose this marker gene, and I'm going to look at the amount of distance between various marker genes, and I'm going various taxes marker genes, and I'm going to say, aha, this is how far you are apart. And I'm going to use this to define things. So we could use the 16S gene, the 16S rRNA for bacteria, and the ITS for fungi. So that's great. This is totally plausible. But how are we going to calculate a sequence distance? First off, which sequence are we going to use? Because we can only use 16S for bacteria for the most part. For different domains of life, we need to use different genes. And how best are we going to capture molecular evolution? So the traditional answer to all of you who come from biology is to align the two sequences and count the number of distances. Count the distance, the number of changes between the two. The problem with alignment distance is that it's awfully slow to calculate. You have to do a nice big matrix multiplication to do this. And it's often quite meaningless. So it's said generally around 80%, so the alignment metric becomes useless. So that is, if you can't align sequences to approximately 80%, you can't really tell any difference any further. So some people might say, okay, 80% is the most we can tell. We're going to define a kingdom or domain of life at 80%. I think that's a rather bad answer because you frequently, if you compare two fungal ITS sequences, you're going to run into greater than 80% difference. Another problem is that alignment runs into problems with structural variation. So that means if I have some sort of mutation that inserts or deletes a whole bunch of bases in this marker gene, that's really going to confuse my alignment and cause a lot of problems and make the distance appear inflated. So that's really a problem as well. Yeah?
What do you mean by alignment distance? Do you have few references that you have a distance?
Right. So if I have two sequences, I can align them and just count the number of changes between them. Yes, one to each other. Yes, exactly. So that's exactly what the last point is, right? Is that if you have an alignment distance, this is just a pairwise distance. It doesn't give you an overall metric that you can place everything into. So it's a pairwise distance, but you can't put microbes into a map. And what we really want is a map of microbial diversity rather than a set of pairwise distances. Does that make sense? Okay, great.
Why do you need a map? I mean, I thought what you wanted was to figure out the features of, or which microbes contribute to some phenotype. Why do you need for that a map?
It's a great question. I think a map is useful for a couple of reasons. First off, if you have a map, it's much faster to do operations on all of it. Secondly, instead of doing pairwise prediction, what you can do is you can plot features, you can do prediction over the entire map. So you could then ask yourself the question, okay, who is a methylotrope across the entire span of bacteria? And can I color a portion of my map that corresponds to methylotropes? This makes it much easier to predict where to source microbes and where to pull things out of your shrimp collection. Okay, great. Yes?
So what you're doing is you take two microbes, you look at them and say they're near each other.
Yeah.
And therefore, this one deals with cold infection, so with the nearby guy.
That's one way you could use this information. I might argue with you that you might want to go down to the gene level and say, this guy we know has this gene is hypothesized to be important. Let's predict who else has that gene and spring them.
I wonder if you cycle it and say, well, I want to know, in order to decide what kind of clustering I'm going to do or how to break these things, but I find two things that both affect, hot and cold. Say, well, they have to be near each other at the end. So whatever my metric is, that's how they have to work.
Right. So you could do a phenotypic clustering.
Yeah.
That's an interesting idea. So the problem you'd run into there is that there's an awful lot of convergent evolution, and there's a lot of horizontal gene transfer. So these would distort your map in two different ways. When bacteria or even fungi share genes with one another, you're going to take 2 points that should be very far away on the map and pull them close together because they share that phenotype. So that's a potential distortion. The other one is convergent evolution. It's two things that are very far apart. You might pull too close together. Whereas we know if you look at the vast diversity of life, there's a lot of things that do things very similar to one another. I actually like bringing up examples about megafauna, in this case mammals, about this. If you look at the branches of mammals, they're mostly differentiated evolutionarily by the continent on which they arose after the Cretaceous period. So if you look, for instance, at marsupials, there are or were marsupial versions of pretty much every major mammal that you can think of that looked very similar to them. that were incredibly distantly related to them. Many of them are now dead, however, but the same thing is true, for instance, if you consider all sorts of mouse and rat and vole-like things. A lot of them are much more distantly related from each other than you would think based on simple morphology alone. So there's an awful lot of convergent evolution in the world. And so while we might like a phenotypic metric, we need something robust to that. All right. So another way that we can consider a sequence is to take an analogy to language. So just like sentences are collections of words, sequences are a collection of subsequences or k-mers. We call them k-mers because they're of length k. So what we can do is we can divide up our sequence into every subsequence of length 2, of length 5, of length 6, etc. And we can just overlap these and go down the sequence. So just as words are co-occurring sets of characters that occur across many different sentences or many different books, so are k-mers co-occurring sets of bases that might occur in many different sequences. If we make k arbitrarily long, eventually they're going to be pretty unique. But if we keep k long enough to distinguish things, but short enough that these patterns will be in common, we can create a metric. Now, what's interesting about k-mers is if you look at k-mer frequency in these Amplicon sequences, in these 16S or ITS sequences, what we can find is that k-mer frequencies evolve like nucleotide frequencies if you give it enough time. And there's some nice prior work in the literature around this. Another good thing about k-mers is that they're easy to compute. You can do all sorts of nice hashing around them, and they're easy to compare with one another. Lastly, there's this beautiful analogy to natural language processing and clustering of text.
So what is K? What are we talking about?
K is, in our case, K is 6. But K is some number.
Coding regions or
this is our marker genes. So this is the 16S rRNA in bacteria, or the ITS rRNA in fungi.
So 2 protein, like 2 amino acids.
These are not these are not actually transcribed. These are part of the ribosome. Their length about 600 base pairs. Don't quote me on that. They just are things that are observed have very slow evolving regions for which you can design primers, followed by hypervariable regions which act like a molecular clock.
These are like correlated with the presence of base pairs. Is that what you mean?
Yeah, so you collect collections of base pairs in a row, right? So if I divide a sequence of base pairs, a sequence of nucleotides into words of length k, of words of length 5, for instance, if I have a very GC-rich sequence, I'm going to have a bunch of GC-rich k-mers. So what I'm going to say is if I look at my vector of k-mer frequencies, there's going to be a lot of things like GGGC or CCCG, etc. And there is work showing that you can model k-mer frequency evolution, like nucleotide evolution. So the idea here is that we're going to take a model from topic modeling, so the machine learning part. We're going to take latent Dirichlet allocation, which is a model that allows us, if we are given a corpus of texts, which we know are made up of words, we want to find shared topics across these texts. So if I give you all the books in a library, if I give you all the patents in the US patent database, and I say, I want you to come up with all the different types of patents, all the topics on which people have patents. What you can do is you can run LDA on them, and surely enough, it will spit out the different types of topics, the different categories in which people have patents, without any information about what's really in there. That's solely based on the word frequency. So the idea is that you find shared topics between texts that are differentiated by their word frequencies. So for instance, going back to our question about patents or books, you would expect books about molecular biology to have the word cell a lot in them. And you would expect books about engineering, for instance, to have the word propeller or engine or things like this in them a lot. Now, a topic somewhere between the two of them, like marine biology, might have cell a lot, but it would also have some frequency of the word propeller, but it would be a different frequency. So given enough data, you can in fact infer these topic models over large enough numbers of texts, and you can infer what these topics are. So just as each text would be comprised of set topic proportions, we can find a sequence we can define a sequence as being composed of sequence topics. So we're going to take a corpus of sequences. These are made up of our k-mers. We're going to find shared sequence topics that are differentiated by their k-mer frequencies, which we know mirror molecular evolution at some scale. And we then say that each sequence is comprised of these sequence topic proportions. So why would we do this? First off, this is a fast, efficient, well-studied algorithm. You can give this an arbitrary number of sequences and it will spit out an answer pretty damn fast. Secondly, the topics in LDA give you a latent space. These give us the coordinates for our map. So if we told LDA to model all of our bacterial sequences with 2 topics, we would have a 2-dimensional map of microbial diversity based off of these topic frequencies. As it turns out, the model would not do so well if you only gave it 2 topics to find, because there's a vast amount of microbial diversity that it could not be modeled. So we need to figure out what the correct number of topics, the correct number of dimensions of this hidden space in which we're going to map everything. So this is going to be much greater than 2 or 3. So here the analogy to a map breaks down, unless you like to think of your maps in 800-dimensional space.
I'm sorry, just a detail, but I'm going to get stuck.
Yeah.
You're dividing the structure sequencing, like the 16S,
Yeah.
non-overlapping k-mers, or you're
No,
sliding?
overlapping things. We're letting things slide.
Okay.
Yes. So yes, you do find correlations between those, but I'm going to make the argument that when you have enough sequences, this isn't so important anymore. LDA is very good at modeling correlations between k-mer frequencies, which is part of the reasons that it's actually a good model here. Because if you look at some model that's going to give you orthogonal components, it's not going to do so well because so many of these are so tightly correlated with one another. Yes?
So if you push the analogy with natural language processing,
Yeah.
if you're going to search the patent database, actually you do a lot better if you look at syntax and build an ecology and extract meaning. Do you have that in the k-mers?
If I gave it a sequence that I thought was meaningful, because okay. So the question was, if I added syntax and some notion of meaning into my modeling of text, I could better extract topics from it. And the question then is, could you do this with sequence? So I'm going to make 2 comments about this. First off, yes, that's correct, but it would be much slower and a more complicated model. Secondly, this is something that you could do if you didn't have marker gene sequences, right? These marker gene sequences aren't actually particularly useful sequences because they're meaningless. That's why they have this nice clock-like evolution, is these hypervariable regions don't really mean anything. And I believe the ITS isn't even transcribed. That's why. ITS, that's what. It's a region between 2 things that are transcribed. So if you wanted to do this on gene sequences, however, where we know something about the syntax, or we can learn something about the syntax, then yes, the point you make is a very good one. And if we wanted to do this, for instance, on a whole genome sequence, yes, that might be something you'd want to do. And that would point that function. Yes?
So but you don't have like some layer of like hidden markers. You know, I'm going to look at this. It reminds me of the system we do recommendations.
Mm-hmm.
Right? We've got a big pile of books, and this book, these 3 books work well in the opposite part, the 7 books work well in the fungus part. And we have a hidden vectors. So I'm wondering if particular sequences here, water is matter in the sequence, this sequence is equivalent to that sequence. Can you actually tell, is the sequence determine it, or would you have to know a lot more structural information?
Right, because these marker gene sequences aren't transcribed, they really don't determine function much whatsoever. So again, this is something that we're going to use to represent microbial diversity and then try to learn from this, because we're hoping that this is an accurate representation of evolutionary distance between strains. Okay. So more formally, I'm a physicist, so I wanted to make sure that I had some math of some form on this. The model in latent Dirichlet allocation, if you look for this is a plate diagram of it, but it's actually not too complicated. The basic idea is here, if we choose a topic, we choose a topic, we put it into the text, and from each topic, we choose a word. The word frequencies come from this word distribution. Each topic has its own set of word frequencies. So if we then plot each microbe on this space in terms of, we then plot each microbe in this latent space. So in terms of which topics comprise its marker gene sequence, what its fractions are, we can now say that microbes are now coordinates in this latent space, and in some sense, probability distributions over these topics. The distance between microbes is then a distance between probability distributions. And we have a good way of comparing distance there, which is the Jensen-Shannon divergence.
I'm sorry, I guess I mean a topic is a set of k-mers or
yes.
probabilities?
Yes, a topic is a set of probabilities of k-mers.
I see, and the topics themselves,
yes.
so a microbe is a distribution
over topics. Right.
Topics.
So right. Just as a text is a collection of topics, each of which is a collection of various word frequencies. So the key question about the number of topics is we want to use as many topics as are supported by the data, which represents diversity. So what we find is that when we run this on fungal sequences, we can take every public fungal ITS sequence that we can download, and we can take a bunch from the Indigo collection. We can find that we can represent all of the fungi with about 145 topics. Interestingly, if you take only the public ones, there are about 80 topics that are needed to model them. And if you take just the Indigo ones, there are about 80 topics needed to model them. But the combination of the two of them requires only 145. So what this is telling you is that there is actually pretty little overlap between those two collections, and that this is kind of a Bayesian non-parametric representation of diversity, molecular diversity. For bacteria, we actually have way more public sequences, and we need about 450 topics to accurately model what's going on. So I have a couple of pictures here that this actually works. On the left side, this is bacteria. On the right side, this is fungi. But on the x-axis here, you see the alignment distance. Now remember, anything to the left of 80% is pretty much unreliable in terms of alignment distance. And on the y-axis, you have this distance, or similarity as calculated in this topic space. What you can see is the vast majority of points lie on this diagonal, corresponding to a relationship of close to 1 between the two of them. In fact, we sort of see this nice curve here where it cuts down around 80%. So what this is saying is that our new k-mer frequency metric in topic space is capturing the differences in alignment distance. You can represent it well, and it cuts off right about where you expect alignment distance to start cutting off.
Now, this is using only the sequence from the 16S or whatever
yes.
you have.
Yes.
But if you take some of those organisms that are fully sequenced and you use basically the evolutionary tree that people, like the phylogeny versus your, the topic distance, how do those compare?
Let me get to the next slide. That might answer part of your question. So what we can do is we can train a nearest neighbors classifier on this latent space, and we can predict taxonomy. And so what we find first off is that when you add more topics up to a set amount, you get improved classification. But we can predict the species level at fungi to about 96% accuracy, which is kind of crazy because I don't think that anybody who's an expert in the field of fungal biology would claim that the species labels are 96% accurate. And at the family level, we have close to as good accuracy as you get, about 97%. With the bacteria, we get really good performance by the time you get to the genus level and so on. Now, notably, microbial taxonomy is always changing. So I don't take these species level results too seriously. The fact that we're getting nice clustering and prediction by genus shows us that we're actually learning something here. If you look at the number of components or topics per strain, You can see that each strain is comprised of around 30 topics. The ones that are comprised of many fewer ones tend to be strains that are in really undersampled regions where we don't know much about them. So there's this new phylum of bacteria that's recently discovered, the Verruca microbia. These are down here. These things all the way over here are really well-studied regions where you need a lot of different topics to truly differentiate them. Our components actually have this sort of interesting distribution of entropy. So when you think about a component, when you think about a topic, if you learn nothing, the topic is going to give equal frequency to every word, every k-mer. So that means it's going to be of really, really high entropy. So these topics here are basically null. They've basically not been counted, but they appear as artifacts when you run the model.
A component is a topic?
A component is a topic, yes, that's correct. And so what you can see here is we sort of have two groups here. There's things that have a decent loading for a lot of different strains, and there's some over here that have, decent loading for a lot of different k-mers, and some over here that are very tightly tied to a couple of k-mers. So these correspond to different features in our latent space. The ones that are high usage and have relatively high entropy are differentiating broad groups of phyla. So if you want to differentiate the two main groups of fungi, you'll find that the two major phyla, the Basidiomycota, are differentiated from the Ascomycota by one of these topics that's in fact a very, very high entropy. The reason it's of high entropy is it's capturing subtle differences in the proportion of frequencies of k-mers. Now these low entropy topics are of low usage, and they differentiate specific clades. So these pull out really weird clades of bacteria. There's one that we found that was predicting in the fungal space a very specific group, for instance, of the Sordaria mycetes, etc. So if we look at the distribution of these components, we can do some type of phenotypic prediction with a metric. And we can do this overall. So because this is the deep learning and production meetup, what do we mean by production here? What we can make a list of here is we can take from 16S or ITS sequence, we can predict any type of phenotype of interest, from assay results to experimental results to presence absence of gene or certain growth conditions, etc. We take this phenotype, we do this latent transformation. This is just shown in 2 dimensions, not 140. And you can color these things, for instance, by phenotype, and you can predict phenotype on strains you don't know that much about. So if we have a phenotype of interest, we can mark known examples in this latent space. We can predict for all the others, and we can use this information to nominate strains for testing, to cluster strains phenotypically, like you were suggesting before. And so really this is the basis of how we do machine learning at Indigo and how we use machine learning on genomics to accelerate our R&D platform. With that, any questions? Yes.
So instead of using modeling,
yeah.
you like things like deep learning that determines embedding coding.
Yes. So I think the clear answer from here is to move to something like deep metric embedding, because you could do that in some kind of supervised context, which would help get you away from the completely unsupervised nature of this. That's definitely what you would want to do, especially if you had whole genome sequences, because the frequencies of various k-mers are going to vary over the genome. Different functional sequences will be tightly conserved, and that's going to really lead to weird spikes in your k-mer frequencies. You're also going to run into this AT versus GC rich region problem, and that leads to problems. So if you do something like a deep metric embedding, something else from a neural network, you can totally do that. And that's something.
Plus, you also use this the relationship of genome distance, right? Because beyond 6 million, you're not going to see the relationship between genes which are positive, right?
Right, exactly. So yeah, so that's a very good point, which is that you want to capture, you might want to capture really long distance correlations between base pairs if you're looking at functional regions. I would claim in the 16S region, which is, or the ITS region, which supposedly evolved neutrally, you're not going to worry too much about that. Yes?
Could you tell us a little more about the computing infrastructure that you guys work with? You know, are you using a particular framework for heavy computational computation and how important is parallelization and those kinds of things?
Okay, so because we're using something relatively simple here, the idea may be a little naughty, but you know, LDA is very easy to parallelize, so we can just honestly use scikit-learn on AWS, which is really fast and really easy to use, which is what keeps us using things like this. So yeah. Just like linear regression is a shockingly useful machine learning model, you know, LDA is shockingly useful in all sorts of weird contexts once you can reformulate your problem the right way. So I'm sorry I don't have a more interesting answer for you there, at least in this terms of this problem. Any other questions? Yeah.
Sorry, I think I missed how you determine the number of topics.
Right. So how do we determine the number of topics? So there's various ways that you can go about doing this. You can look at the likelihood or something like this. What you can also do is, I'd ideally want to fit this where the number of topics is chosen from a Dirichlet process instead of a Dirichlet distribution. I can't really do this. The fast algorithms don't really do that, and I don't want to do sampling. So what's commonly done is you fit a variable number of topics, and then you look at where the number of topics that you fit sort of flattens out, so you stop fitting more topics. You know, if you give an LDA 1,000 topics and there are only 100 or so true topics in it, you're still only going to find about 100 true topics in it. So that's how we can narrow this down, is we can gradually increase the number of topics until we find a representation that's stable.
So you know that the true topics is there something that you know that it's a number of true topics?
We infer it. We see where the number of topics inferred stabilizes, basically. We say that's going to be the number of true topics. But yes, there are probably more principled ways of doing this. On some level, we want to maximize predictive accuracy, and you can also, you could give it, say, a predictive task, like classifying, based on nearest neighbors, classifying at the genus or the family level, and you can see where that flattens out. Empirically, I can state that we're about it where it flattens out for that sort of predictive test as well. Yes, yes, you could totally, yes, that's exactly it.
Is there a disadvantage to using too many topics?
Yeah, in the sense that as you increase the number of topics, you're increasing the dimension of your latent space, so computation just becomes slower.
I have a follow-up question in terms of the technical side of things. I'm wondering why it is that you were saying that it's sort of limited the way that you use AWS and sorts of learning algorithms that you run on AWS. Is that because the operation doesn't need anything more advanced, or is it?
It's just because we don't need anything more advanced right now. We have other deep learning models that we are testing for other uses, such as functional prediction and these sorts of things. You know, we run on AWS with GPU instances and the like.
Do you think that you might expand to more advanced?
Yes. Yes. If we wanted to turn this into something like deep metric embedding, then we would need to do something considerably more advanced architecturally. Okay, great. Thanks, everyone.
Thank you so much. I find it very interesting, and I think we have a good crowd here. That's it, everybody. The coffee and the beer is complimentary. We work is paying, not me. So feel free, and good night.